When I think about metabolism, the first picture that came in my mind is this beautiful and complex network of reactions to build and destroy some compound in the cell. But the question is, how emerged this complex network? We have many hypotheses, and I will present that my hypothesis. And this hypothesis was published in the last Friday in the Biosystem Journal. So, in the first part, I will talk about the two prebiotic chemistry that I call prebiotic chemistry and the protobiotic chemistry as a scenario to restructure the metabolism. The prebiotic chemistry is the traditional prebiotic chemistry. We talked about this and attempt to identify the conditions in the early Earth that led to the formation of the building blocks of life. And evidently, we have many demonstrations that it's possible and synthesize riboses and nucleotide base, small peptides, carbohydrates, lipids, and many other compounds important to the cell or to the life. But the prebiotic reactions were important to increase the availability of building blocks. But when we look for the prebiotic reaction, where these reactions were not incorporated in the biological system. For example, here, when we're looking for this example in Sutherland and group, these reactions to synthesize the pyrimidine ribonucleotides. Here we have a sequence of reactions. It's totally different of the way that the cell used to produce the same compound. For the other side, you have the protobiotic chemistry. That is we will refer as protobiotic chemistry. That's a word that are trying to discover metabolic hypercycles operated in prebiotic Earth that happen without the need of biological catalysts. Okay, but this set of reactions that possible occurred on the prebiotic Earth without the aid of the biological catalyst, but these reactions were further incorporated in the metabolism of the biological system. And we suggest that we had a sequence of reactions, and in this case, only with the chemical catalyst. And with the emergence of the peptide replaced this inorganic catalyst and structured in this way, incorporated this route in the biological system. We have, for example, this work of the Markhausen group. Here we have the modern glycolysis and pentose phosphate pathway. And we have many simulations of these same pathways without any enzyme. And as you can see, it's very similar, the reaction that the group obtained only with the iron as a catalyst and the biological pathway that was working in the biological system. Evidently, when we talk about the origin of the metabolism, we need to put this origin in two scenarios, in the RNA world and the ribonucleoprotein world. The RNA world, everybody knows, it's the most popular theory about the origin of life, and proposed that we had at the beginning polymerization of small RNAs that had the capacity to self-replication. And the next stage of this evolutionary process, this simple replication sophisticated. And at this point, you have the ribozyme activity, the polymerization and ligase, all in these two. We need just this reactions. And at the end, in the next step, we had the regeneration of food that supply the replication. In this case, the initial biological synthesis of the ribonucleotide, and emerged a new activity, nucleotide phosphoribosyltransferase transferase. And the last step here in the RNA world is the emergence of the ribosome. The emergence of the ribosome made possible the transition between the RNA world to ribonucleoprotein world. At this point, the RNA established a crosstalking with the protein world. And the problem here is how occurred the emergence, the transference of this property, these activities that initially the ribosome do to the protein. Thomas Cech have a model, and in this model we have ribosome. This ribosome initiates an interaction with some peptides, and at the end of the process, the catalytic functions transfer to the peptide compound, or like an enzyme, or keep it as an coenzyme or ribonucleoprotein. We have many examples of this when the ribosome is a ribonucleoprotein. That's very important in this moment. So, the question is, what was the evolutionary force in the RNA world? And I have two questions. What are the biological functions that you can see in the RNA world? And replication catalysis. What was the selective pressure to structure the RNA world? The selective pressure was to replicate the RNA. In this case, the replication was the selective pressure. But what's the pressure to structure the metabolism in the RNA world? Synthesis of nucleotides and polynucleotides. Okay. When you look for the ribozymes that have the capacity of do some steps of the reactions, we can find that one very interesting reaction that can be done by a ribozyme, but in this case, it's the synthesis of the nucleotide, but in this case, this ribosome just put together pentose phosphate and one base. And again, when you look to these reactions, it's totally different the reactions in the biological system to synthesize the same nucleotide. Well, the first conclusion here is that the metabolism in the RNA world were not incorporated in the biological system. When you look to the RNP world, the main process here is the emergence of the ribosome. The ribosome is a very complex molecule, ribonucleoprotein molecule. And we have the large subunit here, the small subunit here. And this machine have the capacity to pass the information stored in the nucleic acid to the proteins. We have two important sites here. In the large subunit, we have the peptidyltransferase center, that is a catalytic site of the ribosome, is a ribosome. And in the small subunit, we have the decoding site. In this point, the mRNA is attached here, attached there to be read correctly during the process of the translation process. So I will do the same question here. And the evolutionary forces in the RNP world. What are the biological functions? Here we have replication, protein synthesis, and catalysis. Evidently, you can see that it's more complex. What was the selective pressure that structured the RNP world? Replication and protein synthesis. And in this case, what pressure structured the metabolism in RNP world, synthesis of nucleotide, polynucleotide, amino acids, and proteins. And when you're looking in the modern organisms, practically all these compounds are derived from three very important pathways. We have the glycolytic pathway here, and from the glycolytic pathway derivating many amino acids here, and the TCR cycle many other amino acid derivated. So here you have the pathway synthesis derivated from glycolysis and TCR cycle, practically all amino acids used in the biological system. And also derivated from glycolysis, we have the pentose phosphate pathways, and this pathway is very important because supply the synthesis of nucleotide by the ribosyl 5-phosphate. And this compound goes to these other pathways and help to structurate a nucleotide. This is in the modern organism, but it's possible these pathways occur in the prebiotic conditions. Joseph Mohan and the group of Joseph Mohan demonstrated that the TCR cycle is possible for without any enzyme in prebiotic conditions. No? And this is a very important cycle to supply the system of amino acids. And again, this picture, the group of Markenhausen demonstrated that it's possible structured the glycolysis pathways and the phosphate pentose phosphate pathways without enzyme. So these two important pathways is possible that occur in the prebiotic conditions before the emergence of the enzymes. So the model that we are proposing, we have this schematic model. We had prebiotic compounds. From these prebiotic compounds emerged the proto-translation system. With this, it's possible the synthesis of the first peptides, and the first function of this, the first peptide was stabilization of the system, to stabilize the system. In this case, binding in the ribosome or proto-ribosome to stabilize. With the complexification of the system, the new peptides structured the emergence of the new metabolic pathways, and these pathways supply the translation system of the substrate to this peptide synthesis. So we need to do other very important question. What is the origin of the biological information? And in 2016, we proposed the tRNA core hypothesis to transition from the RNA world to ribonucleoprotein world. In this proposal, we suggested that the tRNAs or proto-tRNAs organized the system, the biological system, and the proto-tRNAs gave origin to the PTC, that's the catalytic site of the ribosome, and also they gave origin to the proto-mRNA. With this, we structured the proto-translation system, and the protoprotein by a positive feedback binding the tRNAs and stabilize the system. So we have three elements here, and the catalytic site of the ribosome, the mRNAs, and the tRNAs that are important, that is important to connect the nucleic acid and the protein world. I have many evidence that suggested that the peptidotransferase had the origin from proto-tRNAs. Okay, we have a similarity between here is an alignment between the modern PTC and ancient PTC constructed based on tRNA ancestral sequence. And when you look at similarity, we have a similarity around 52% in the primary sequence. When you look from this ancestral sequence, when you reconstructed the three-dimensional structure, you can see here, and make a superimposition with the modern PTC, we have a similarity around, let's see here, 92%. It's practically similar. And this is very interesting because the group of Adayana suggests that this is the first part of the ribosome, included. She say this that the ribosome is ancient machine embedded in the contemporary ribosome, and recently, other group constructed a synthetic PTC that had capacity to formate a peptide bond. This is very interesting. And the very interesting here is this had capacity. When I look for this region of the ribosome, is exactly the region that I found similarity with the tRNAs. Exactly the same. So the other part of this complex history is the origin of the first genes. If you have a ribosome, we need genes. And in 2000, we published this paper. And this paper, we reconstructed the ancestral sequence for each kind of tRNAs and put three by three together like a gene, right? And compare with the modern proteins. For our surprise, when we look to the results, we had similarity between this ancestral tRNA sequence with the main proteins of the glycolysis and gluconeogenesis pathways. From this primary sequence, we have constructed the three-dimensional structure. Here you can see the enzymes and names of enzymes. And the first analysis, we can see here sequential steps of evolution. It's possible to make more restricted analysis, and in this way you have a very ancient structure. Here is the second step of the evolution of this protein. Here's the third step in red, the first step, in green, the second step, in blue, the third thing, in yellow is the modern protein. So when you look at this picture and this table, here is the enzymes. The data from the red structures are here. The data from the blue structures are here. And evidently, you can see from here to here, the structure had a process of complexification. When you look to the most ancient structure, the structure binding nucleic acid, guanosine triphosphate, nucleic acid, as the model suggests. So when this structure incorporates many other parts, this structure, this protein change your affinity. And when you look at this structure in blue and the protein substrate here, we start to see the modern substrate. So we suggested that we had many nucleotides here that formed the first tRNA, and the first RNA in this case is the tRNA, structurated the primitive ribosome that was supplied by the amino acids, and the other proto-RNAs worked as mRNA, it was possible the synthesis of the first peptide in coding. Here emerged the genetic codes, a new property of the biological system. And this protein start the catalysis process. And these RNPO, as we suggest that occur like an open system without membrane. After the emergence of the lipids, this system was encapsulated in the cell, in the first cell or in the proper cell. So this picture, these peptides that emerged of the primitive translation systems replaced the inorganic catalysts that are present in the environment. For example, here we had pathways, prebiotic pathways, non-enzymatic reaction, non-enzymatic. With the emergence of the peptides are faded some spaces that before are occupied by the iron or any other compound. So some ideas about this. The data suggests that the first genes and tRNAs had a common origin. The first encoded peptides did not have the catalytic function, just binding the RNAs. It's very important. When we reconstructed the ancestral structure, this part of the structure always is the catalytic site. So the catalytic site must be the primitive part of the proteins. Proteins were structured from the catalytic site by an accretion mechanism. Studies suggest that the citric acid cycle and glycolytic pathways could occur without proteins in a prebiotic environment. Several pathways of amino acid synthesis are derived from these two cycles, the CR cycle and glycolytic pathways. This way, it suggests that with the establishment of the translation system, protein replaced the chemical catalysts, increased the efficiency of the citric acid cycle and glycolytic pathways, and thus increased the availability of amino acids for the translation system. So this is my collaborators. Thank you. I'm open to questions.